Dear ladies and gentlemen, my name is Oleksii Tyshchenko and I represent the University of Ostrava. So my current talk will have to do with the GEOCLUS, a fuzzy-based learning algorithm for clustering expression datasets. First of all, I'd like to introduce the talk structure for today. So first of all, I will provide some introduction to the problem statement, to the problem area. Secondly, I will uh, announce the motivation of the current research. Thirdly, I will provide the description of the introduced procedure. Then I'll show the simulation comparison details for the obtained results. In the final part of my speech, I will provide a short summary for the announced research. So DNA microarray experiments monitor the expression levels of many genes together. A considerable amount of raw gene expression data is procured from these experiments. Clustering algorithms mostly discover hidden patterns and natural grouping for homogeneous structures and substructures of biological significance from an expression dataset. In most cases, gene expression datasets have an intricate high-dimensional data structure associated with imprecise information. Usually, these structures contain lots of noise. So one of the dominating challenges of clustering gene expression data is the big P, small n problem, which means that a scenario is characterized by a prominent attribute or feature size in comparison with the sample size. The expression profiles of the prognostic clusters in a gene expression datasets are quite similar. The boundaries between the classes at the same time are quite vague. Speaking of the proposed novelty, I should admit that the introduced approach helps project the microarray data onto the Riemann manifold with the Fisher-Rao information metric. Manifolds with the Riemannian matrix maintain intrinsic characteristics of the original space. Data is mapped to a hypersphere, at the same time keeping all these straightforward intuitions and keeping all the local nonlinear interactions between the features. The method is mostly built on the fuzzy semidoid algorithm, and the biggest benefit of this semidoid procedure is that a midoid is used as a cluster representative, and the usual preference is the cluster mean. And like GEOCLUS favors the geometrical median as a midoid and a more robust central representative for datasets with noise. As a distance, we use the geodesics metric, and geodesics on the hypersphere correspond to line segments in space R. So speaking of the motivation of this research, I should admit that we would like to develop an efficient clustering procedure for cancer patient stratification. And so, so mostly we would like to introduce a robust fuzzy-based clustering technique which searches for treatment groups in the cancer datasets. It mostly partitions cancer samples with distinct histopathological characteristics. Multiple genes impact a disease state through nonlinear interactions, and a single gene usually doesn't contribute a lot to the disease state. So the correlation among expressions is also well documented. So these two observations nurture the idea that gene expressions can be transformed to a low-dimensional space that captures the variations in the data structure. While usually gene expressions are high-dimensional directional data, and like the prior research suggests that a spherical space or a positive curvature space be a more potent choice for assessing this resemblance. So speaking of mapping the expression data sets on the Riemannian manifold, we should admit that we come to a high-dimensional expression data set. So as you can see, like we have a matrix that contains like where row vectors represent the samples taken under consideration, and like here you can see that we have some mathematical background for the introduced technique. We provide some like mathematical background for the future research, and like uh, so where you can see that we have some we introduce the m-1 dimensional hypersphere with coordinates, where we provide the mathematical background for these coordinates. At the same time, we also introduce the geodesics um, preliminaries for the hypersphere, and like we also provide uh, the way how we can calculate the geodesic distance on the hypersphere between two feature vectors a and b. So everything is calculated according to the expression 1, where r is the radius of the hypersphere. So speaking of the local information parameter, it's associated with each feature vector xl. So the local information parameter is determined according to the expression 2. And we also introduce the threshold distance, which is based on a standard deviation of the attributes. And the threshold distance is defined according to the expression 3. And you can see the explanations for all the required parameters for this expression in the lower part of this slide. Well, generally speaking, speaking of the geodesic fuzzy clustering technique, we should say that it's based on the minimization of the functional 4, having restrictions expressed by the formula 5. And actually, distance is measured according to the geodesics metric. So this distance captures local and nonlinear interactions among the features, between features, and it's defined according to the formula you can see in the lower part of this slide. And like some minimization of the functional for results in new calculations of the membership functions. So now we actually move on to the practical part of our research. So we use 4 data sets that have to do with different sorts of cancer. So as you can see, the first data set, GEC 2034, had to do with lymph node negative breast cancer. The next dataset, GEC 129617, had to do with the ovarian clear cell carcinoma. The third dataset had to do with the prostate cancer. The fourth dataset had to do with the brain cancer patients. So at this slide, you can see some data characteristics or of the previously mentioned datasets. So now we move on to the practical part. Now we can see some graphs here, and I should also say additionally that the current research had a vast pre-processing stage. So basically, we use the principal component analysis, like the so-called PCA technique, um, to clean the data and to project data on low-dimensional space while maintaining the intrinsic data characteristics. Here, data is projected on 3-dimensional space. So GeoCLUS partitions datasets on the hypersphere actually. So here we use, just as an example, only the dataset GEC 15484, and actually you can see different cases for the same datasets. So our approach embeds the microarray datasets on the remaining manifold H plus, and H plus is the positive orthant of the hypersphere with the radius 2. So the threshold distance TD is calculated according to the equation 3. And actually, during the experimental part, we had different values for the user-defined parameter nu, as well as different parameters for the calculated threshold parameter TD. The local information assigns higher weights to outliers in the dataset, and GEOCLUS clustered the datasets with iterative minimization of the functional 4. So the fuzzy parameter m was chosen to be 2 across all the experimental part. So here you can see like the performance evaluation. In our research, we used 4 performance metrics such as classification rate, silhouette index, the Davis-Baldwin index, as well as the Zelinsky-Karabash index. Um, Classification rate measures the accuracy of the learning algorithm. The remaining 3 performance metrics are internal measures that depend on information from the feature vectors in the data set. Actually, the best values are marked with bold, so you can see which algorithm shows the best performance for each case. So for every data set, we use 3 different techniques: K-means, FCM, and GEOCLUS. And finally, you can see the required parameters for each method, and you can see the best result. So based on these results, so we should say that the GEOCLUS has the optimal silhouette and Davis-Baldwin scores for all the data sets except for GSC 50161. The fact that GEOCLUS had the highest classification rate score for all the data sets indicates Go has a well-separated cluster structures. So summarizing the research, we should say that a robust fuzzy-based clustering procedure based on the fuzzy simidoids algorithm for cancer patient stratification was offered. Uh, so the introduced techniques is mostly focused on microarray data sets, and the clustering process occurs in the curvature space. The obtained results were compared with the generic k-means and with the FCM algorithm in terms of classification rate on the microarray data sets. So we used 4 different techniques, 4 different metrics for assessing the cluster structure obtained during parsing the data. The experimental findings indicate that GEOCLUS can be a plausible tool for clustering expression data sets, and the proposed approach partitions the data into distinct groups for further analysis. So thanks a lot for your attention. That's all for today.